I'll go ahead and introduce Dr. Debanu Das. It's my pleasure to introduce him to present today. And as Sam said, we're swapping roles. So as a bio, Dr. Das has over 20 years of experience in high-throughput protein X-ray crystallography, structure and fragment-based drug discovery, and structural biology. He's the co-founder and CEO at Accelero Biostructures and Exposed Therapeutics, both of which are based in SF, here in the Bay Area. And Accelero Biostructures specializes in high-throughput protein X-ray crystallography-driven drug discovery, it's a mouthful, platforms. And Exposed Therapeutics is focused on advancing new oncology therapeutics based around the DNA damage response. Prior to his current roles, he served as staff scientist in the Structural Genomics Division at SSRL, which is the Stanford Synchrotron Radiation Light Source, another mouthful, at SLAC National Accelerator Lab in Menlo Park. He got his postdoc training at Lawrence Berkeley National Lab and the University of California, Berkeley on structural genomics and membrane protein crystallography. He holds an integrated MS in Science and Engineering from the Indian Institute of Technology in Kanpur and received his PhD from Rutgers in New Jersey. He is going to speak with us today about target hit discovery by molecular library screening by crystallography. And with that, I will turn it over to you, Debanu, to take it away.
Thank you, Karen. Can you hear me okay?
Yes.
Is my screen pinned? Sam, is the screen pinned? Can you see the slides?
Yes, we can see it. I guess if you just want to go on presentation mode.
Okay, all right. So Accelero Biostructures is based in San Francisco, and we are a protein crystallography-driven drug discovery company, which means that we try to drive structure-based drug discovery by experimental and empirical determination of high-throughput X-ray crystal structures of our targets. And what I'm going to talk to you about today is doing target hit discovery by library screening directly by X-ray crystallography. Okay, so most of us are familiar with this flowchart of drug discovery. There's a lot of high throughput screening that goes on in the early stages of drug discovery. There are hit to lead stage, then the lead optimization state, candidate seeking, and so on. And many of us who are speaking in this conference today work in this early drug discovery space. Now, what I'm going to talk about is how we're trying to do more to tackle more targets, make more drugs at less time and cost in this early phase, and really driving it by what is called fragment-based drug discovery and combining that with experimental 3D protein structures. Again, in terms of pipeline or where we are in the drug discovery process, it's really at the target ID and hit generation step that we focus on. So fragment-based drug discovery is, or FBDD as it's commonly called, similar to its counterpart structure-based drug discovery, SBDD, is widely used to provide novel leads for developing new therapeutics. And it's a very efficient way of scanning chemical space. So instead of looking at billions of compounds as are done with Dell library screening, or even millions of compounds done by computational screening, or hundreds of thousands of compounds done by high throughput screening, if you look at fragment libraries, you can cover an immense amount of chemical space, novel chemical space, just within about 2,000 to 4,000 compounds, or even less. Even 1,000 compounds will give enough flexibility and diversity to cover chemical space. Fragment-based drug discovery has resulted in about 30 new drugs entering clinical trials, with 4 already in the market. Structure-based drug discovery itself has led to about 200 FDA-approved drugs. Now, the absence of crystal structures of targets bound to the fragments or molecules of interest is a significant impediment to medicinal chemistry optimization. So without really having empirical structures, you rely on computing and simulations and so on. And conventional crystallography is inefficient for scanning a fragment library due to expense and effort. Often due to this, other biophysical methods like surface plasmon resonance, thermal shift assay, or NMR are used to pre-screen for fragments that bind, while protein crystallography is used in a second step to determine structures. Now, what we have done is, with foundational expertise in structural genomics and developing high-throughput protein crystallography, over 20 years now, we have developed novel capabilities where we can directly use X-ray crystallography in an ultra-high throughput mode to directly screen compound libraries by X-ray crystallography, and leading to potentially a 5 to 7 times reduction in early drug discovery times, providing an unprecedented experimental route to high quality, high reliability, and high value results simultaneously, which is the identification and assessment of fragment binding sites and their poses, target ligandability, the differentiation of orthosteric and allosteric sites, and binders. So all this can be done in one step in our ABS one-step platform, as we call it, providing very immediate actionable input for medicinal chemistry and biology to act on the results. In concept, if you look at what fragment-based drug discovery is, so here's a schematic of traditional high-throughput screening where you have a target and you screen it to the library. These libraries often have molecules that are fairly large in size. They bind to the target. Some of them may bind. They're not very efficient at binding. And then you can optimize the compound based on the binding and try to make it more selective. Fragment-based drug discovery uses a different approach. It starts with small molecules, fragments of molecules. So these are essentially parts of bigger molecules, but they're still small molecules. And the goal is that they should bind at either orthosteric, known functional sites or potential allosteric sites. And once they're bound, these are very selective in how they bind. And because of the selectivity, you can then directly enhance them and grow them to make very target-specific molecules. Yeah. And this can be done by either growing a fragment into a space or linking fragments bounded up in nearby spaces and so on. Now, what happens in conventional fragment-based drug discovery is that because of it's the amount of work that is needed to be done, often you do this pre-screening. So this pre-screening can be done by thermal shift assay or differential scanning fluorimetry assay. And then an NMR step can be used to look at interactions and then finally go to try to determine crystal structures at the last step before doing chemistry. However, from biophysics, we know that X-ray crystallography as a method has really the largest interaction detection range. So it can detect very weak interactions to very strong interactions, whereas other methods like NMR and surface plasmon resonance have a much narrower range of detectability of affinities of molecules to their targets. Biochemical assays, in fact, have the least amount of detection range. So you really need a molecule to be fairly potent before it can be described and identified by an enzyme assay. Now, the span of X-ray crystallography really makes it very suitable for fragment-based drug discovery, because fragments, being small in size, initially have very low, weak interactions with the target. And so X-ray crystallography is an ideal method for detecting it compared to other methods. So our goal has been, how do we get directly from a fragment library or molecular library directly to structural information? And that's what forms the basis of our ABS one-step platforms, to go from fragments to structures in a single step. What are the benefits of this approach with our platform? The first benefit is structures first. So we're not playing in the dark with molecules that we do not know where they bind. So essentially, we're getting massive gains from parallelizing and resolving bottlenecks at every step of the structure determination process, producing hits and structures in parallel. And once we have these hits, usually we have multiple hits. The hit finding rate is pretty high because the hit rate is very sensitive. So all hits can immediately advance to biology and chemistry. And the other big thing is that there are no false positives. Unlike biophysical methods, where you might get false positives, there are no false positives, and the hit rate is very high, much higher compared to biochemical assays or biophysical assays. We have developed novel processes that rely on automation and parallelization to quickly resolve bottlenecks. It's an empirical approach, so there's no guesswork involved. And we can do it with high speed, high quality, and high reliability. And what is the outcome? We deliver actionable assets that essentially identify binding hotspots, tell us about target ligandability by looking at binding hotspots, and we can assess whether they are functional site binders or allosteric binders by seeing their structures. Right? No other method tells you about where the molecules are bound on the target other than crystallography. So we're able to explore new targets for suitability to launch drug discovery programs. explore crystal systems for suitability for fragment-based and structure-based drug discovery programs. In the end, ultimately providing us an unprecedented ability to visualize 3D protein fragment ligand structures in a single step. Now, what allows us to do this? Why can we do this now that we couldn't do this before? So there's some background to it. There's a historical perspective that I'd like to quickly go through. So in the late 1990s, human genome sequencing was really getting started at the NIH and at Accelero. And there were a bunch of structural biologists who were very highly respected senior crystallographers proposed, what are we going to do with all these gene sequences once they're going to be sequenced? How do we understand what the proteins are doing, right? And they proposed doing what is called building a structural complement of genome sequencing efforts. So essentially getting structures at high throughput that would tell us what the functions of these novel genes were, the proteins they encode. So this gave rise to the field of structural genomics that we were essentially involved in before Accelero Biostructures. And about 20 years ago, in 1998, Sang-Ho Kim at UC Berkeley, whose lab I essentially did a postdoc in after my PhD, but this was the year I saw her starting my PhD. So he proposed that in this really path-breaking paper at that time, the structure-based assignment of the biochemical function of a hypothetical protein, a test case for structural genomics. And the goal was that if he could make the entire gene-to-structure process all the way from automating high-throughput construct design, cloning, protein expression and purification, protein crystallization, X-ray data collection, structure determination and analysis, we could really bypass trying to characterize novel proteins by biochemical assays, because you cannot essentially build a biochemical assay for a protein whose structure function you do not know. But we can actually assess what their potential functions might be just based on the structure relationships that we can do. So this is what led what we were doing for a long time in what is known as the Protein Structure Initiative, or the Structural Genomics Program, between 2000 and 2015, where everything was automated in parallel. So we were essentially involved in doing all of this end-to-end. Giving us 15 years of high-throughput protein crystallography R&D operation experience at the Berkeley Structural Genomics Center, which Sang-Ho Kim was heading, and later on at SLAC National Lab at the Joint Center for Structural Genomics, which was an industry, academic, government consortium led by Ian Wilson at the Scripps Research Institute. So this experience allowed us to really build a robust pipeline in structural genomics from gene to structure. And we determined 1,500 novel protein structures deposited in the PDB, obtained around 300 publications on structures and methods development that really gave rise to our genesis in Accelero Biostructures, that we transported all that expertise into building up, doing high throughput and ultra high throughput structure-based drug discovery for fragment-based drug discovery and other structure programs. Yeah. So what do we do in this? So what has been the outcomes and successes with our efforts so far? So this is an example of the kinds of targets that we have looked at. Some of them started with initial lab proteins, very simple proteins like trypsin and lysozyme and thaumatin, and then we went into more complex systems, but I'm just showing this for illustrative purposes as to what protein crystals look like of different kinds. And you can see these here. These are these crystals that of different proteins that you can see here as I move my mouse around. But we essentially have now run this hit determination paradigm with this platform on 11 targets so far. So these are various kinds of targets. These are very proprietary targets, so they're just coded. But this gives an idea of the kinds of molecular sizes, library sizes that we've looked at, all the way from as small as 30 compounds to as big as 300 compounds at a time. The platform has been entirely built out now, so we are now at full capacity, where we can screen about 1,000 compounds in a particular week once the assay is set up. This means that we can go directly from dosing the crystals, just like you would dose protein in solution in a biochemical or a biophysical assay with compounds in high throughput. We actually dose crystals with compounds and collect their data and solve their structures in a very rapid process. So this involves a huge amount of parallelization in how we handle crystals, in how we collect data, how we process the data, usually for about 500 to 1,000 crystals at a time. For a whole library screen, we're looking at a few terabytes of data, and then these essentially have to be processed all in parallel. So we have a massive computing system that helps to deal with all the processing. So we can actually go from dosing the crystals. Once the assay is set up, the crystal system is set up, we can go from dosing crystals to understanding if there's a hit or not, all within a week for about 1,000 compounds at a time now. And this gives an idea of some of the kinds of hit rates we have, right? So these are, for example, with lysozyme initially, we had a 4% hit rate, 4 out of 100 data sets gave us structures. Here's 1 out of 50, but these are much higher than what you would have in high-throughput screening. For example, in this case, this protein, we had 20 hits out of 100 data sets, fragments that we tested. In one case, we had 30 structures out of testing 50 fragments. And then we have more recent ones. We have really moved into doing 300 at a time very quickly. Some of these are in progress. Some of these are SARS targets. And then this gives an idea of how we deal with these structures. So we have actually got fully refined structures in most cases, telling us what the interactions are. So as an example, here's the first proof of concept study with lysozyme, right? This is the protein structure, and these are molecules that we found bound to the structures. So you can immediately see how easy it is to look at the validity of hits by doing crystallography directly. You can see exactly how they're bound, what the interactions are to the protein, what the water structure is, and so on, all very important things in actual drug discovery. Here's an example of a proof of concept study with UCSF, where we looked at a DNA methyltransferase. And again, you can see a couple of representative molecules with the electron density there. So electron density doesn't lie, right? So this is why it comes down to the fact that these are really, there's no false positives. You know where they are, what they are doing. Of course, in some cases, there might be hits that are not productive in the sense that they could be bound at lattice interaction sites and not really reflective. But even then, you can quickly eliminate those hits as being unproductive hits. But it's not a false positive that you'll get a hit. Again, this is an example of an MRE11 endonuclease. Again, you can see the kinds of molecule hits that we get on doing a screen like this. This is an example. This was a very nice project for us. In fact, this was with a pharma company, and they had essentially run a bunch of biophysical screens. They gotten some hits with no clues, no idea about where the molecules were binding, what they were doing, what to do, what chemistry, what kind of synthesis to do on them. We essentially, gave them a bunch of structures all at one go by running this screening, right? So we simultaneously obtained hits, their binding locations, their protein ligand interactions. The chemists were able to quickly and effectively plan synthesis strategies, and the biologists could readily follow up with assays and finding out where they are. You can spend months and months trying to optimize your assays to find these hits, but in case you have hits that are not at the known functional site, how do you then measure any biochemical assay with it, right? The assay is not even set up for that. So you can go, spinning your wheels, wheels in trying to follow up with these, and this is where the power of the platform really, or the approach, gives us, allows us to converge very quickly to productive and actionable steps. This is an oncology target. This protein has been studied for many years, maybe about 15 years, and it's very important in oncology. But there are no, and in fact, a bunch of molecules have been published coming from high throughput screening and virtual screening and so on, pharmacophore modeling, and there are no experimental crystal structures of any of these compounds that have been published. So we ran the screen and essentially got the first crystal structures of the target bound with drug-like molecules, simultaneously obtaining hits, their binding locations and interactions, and really discovered and looking at validation of an allosteric site here with bioassays in progress. We, in fact, spun this out into a new company, which I'll get to shortly. So what has been our journey so far in terms of development logistics? We conceptualized this platform, developing this platform in fall 2014, and really the concept was grown out of our foundations in structural genomics and high-throughput X-ray crystallography. And then we started work on this in 2015, about 6 years ago. And we have essentially developed special media for crystallization and robotics protocols, fragment binding, how to handle fragment binding, data collection protocols, these are X-ray data collection protocols, a lot of high-performance computing, data generation and database development for strategic implementations and IP generation. So the platform is essentially an expertise-driven combination of methods, protocols, tools, hardware, and software. We primarily use fragment libraries. However, we ab initio as the first screening step, primary screening as we call it. However, we can also take libraries that are prioritized from biophysical and biochemical screening that may have been done before. So in many cases, what is done in biophysical or biochemical screening or computational screening is that you end up with maybe 30 or 40 or 50 molecules to move to crystallography, and we are saying, why do we need to stop at 50? If you're doing 100,000 compounds by computational screening and you get 300 compounds, why do only 30 by crystallography? We can do the entire 300 by crystallography. So that's one of the advantages as well in terms of using this. As I said, we are now up to doing pretty ultra high throughput, as we call it. In a latest development in January this year, we have actually started screening more than a fragment library. We have moved into screening a scaffold library, which are larger molecular sizes from fragments and based on fragment hits for a target. So we're now really calling it a screening molecular libraries, as I've defined in the topic for this, in the title for this topic. And again, we did a spin-off of Exposed Therapeutics in fall 2019 using results from this platform. I have maybe a few minutes left, I guess, or maybe 10 minutes left, and I have a few more slides, so well in time, I think. So I'm going to take you through a bit of the journey of Exposed Therapeutics. So Exposed Therapeutics is also based here, and what we're doing there is using a cutting-edge approach, and this cutting-edge approach is our platform, really, to develop target-specific cancer therapeutics against DNA damage response proteins, DDR. So DDR has become very popular over the last few years. We started, in fact, this project that I described in 2015. At that time, there was no company doing DNA damage response, almost no company. Only one looking at it tangentially. And then in fall 2015, the Nobel Prize for DNA repair was awarded. And over the last 5 years, that led to creation of many companies. The other big factor was PARP inhibitors. These were brought to the market by AstraZeneca, and that really gave a boost, both the combination of PARP inhibitors being in the market as well as the Nobel Prize really gave a boost to the field, and there are about 15 companies now looking at in the DNA damage response space. So what is the problem here that we're looking at in DNA repair? So there are roughly 5,000 new cancer cases reported per day in the US alone, and there are roughly 600,000 deaths per year arising from cancer-related complications. So it remains a very unmet medical need, and there's a need for new treatment modalities. Now, this is our DNA, of course, a schematic of our DNA, and it is known that many cancers arise from inherited or sporadic defects in DNA damage response, providing a unique targeting opportunity. So you can see that there are several factors that cause DNA damage. There are endogenous factors like replication stress, cell metabolism, there are oxygen radicals, and then there are exogenous factors like radiation, viral infection, chemotherapy, and so on. And they give rise to a bunch of different ways of DNA damage, which are then are repaired in the body by various mechanisms that are acting all the time. And for example, mismatch repair, nucleotide excision repair, base excision repair, translesion synthesis, homologous recombination, non-homologous end joining. So there are various ways by which our body is normally tackling and repairing the onslaught of these damages that occur all the time. So what we are building at Exposed Therapeutics is a DNA damage repair platform. The core of the platform is high throughput X-ray crystallography that I just described, screening libraries that using Accelero Biostructures platform. And then that is then coupled with biochemical assays and biophysical characterization and taking inputs from various sources, primarily, again, for proprietary fragment libraries, but also we can take hits that are emanating from other methods that may have been reported, and really driving this process to feed into medicinal and computational chemistry, then cell-based assays, lead optimization, and then followed by in vivo characterization. So that's really the DNA repair platform that we are building. Now, this, of course, cycle is this kind of a schematic is common to almost any modern drug discovery program. The unique part of our DDR platform being that this is focused around DDR targets. It is using high throughput X-ray crystallography as a hit generation method, and pretty much those are the main features or differentiators of this platform. All right, so a couple of examples of what we've been doing, and we published this recently in a paper in October: fragment-based and structure-based drug discovery for DNA damage response proteins. This is one of our programs. This protein is highly expressed in rapidly replicating cancer cells. It's called XTX1. It's our target XTX1. The protein is actually APE1 AP endonuclease, and it has broad applicability. Targeting this has broad applicability, including in prostate cancer, lung cancer, ovarian cancer, breast, cervical, pancreatic, colon cancer, and leukemia. So DNA damage response is prevalent in almost all cells. So what it's doing is that in these cancer cells, it's repairing cancer cells, right? So it repairs cancer cells at a much faster rate than it repairs normal cells. So the whole goal is to be able to stop that from happening, to reduce repairing of cancer cells and ultimately leading to their death. It's particularly relevant in glioblastomas, which has been, a really big refractory case to solve, as was discussed yesterday in several talks. So we at Expos have now, working on the first drug-like small molecules to look at this. We're also looking at two approaches simultaneously, both the small molecule inhibitor approach, SMI as we call it, and targeted protein degradation, or the PROTAC approach. Again, these are some nice crystals of this protein, and it's an illustrative example of one of our hits, partial hits, that we have bound to the protein. Yeah? All right, I think I'm on my last slide now. Let's see. Seems my last slide is not showing here. Okay, I don't know why my slide 20 and 21 are not showing up. It's very strange. Are you all there? Can you hear me?
Yeah, we can hear you.
Okay, let me try to find my last slide. All right, that's very strange. Anyway, so my last slide was another example of what we were doing, another polymerase that we're looking at in translation synthesis, and I was going to show you a similar illustration of what the crystals look like and what the electron density looked like of the hit, but it's not showing up on my slide. So I'm going to stop there, in fact, and happy to take questions.
Wonderful. Thank you again, Dr. Bano, for your insightful presentation and showcasing your new group, Expos Therapeutics. And if there's any questions from the audience and from the chair.
Yeah, so I want to also say thank you. That was a really excellent presentation, and congrats on the new company, or fairly new company. And I have one question about the use of the X-ray crystallography system in terms of what you mentioned about, otherwise you don't know which biochemical assays to use necessarily. So I guess my question is, can you use the information you get from ABS one step to not only run the right biochemical assays, but maybe fewer of them as a result of the knowledge that you've gained from using your technology?
Yeah, for sure. So, there is in this whole screening world, that this has been demonstrated and published multiple times, that these orthogonal assays, if you do biochemical, biophysics, even between biophysics, between NMR, thermal shift, and SPR, the overlap of hits is not great. Uh, and so companies choose to do orthogonal assays and then try to find the intersectors that they can move forward. But in most cases, or in many cases, they don't overlap. There is some correlation seen between NMR and SPR. They tend to correlate a bit, but again, it's very target dependent as well, depending on how what the target properties are and so on. This whole ambiguity goes away with doing crystallography. If of course, on the assumption that crystals can be grown for that system. So not every target system will be amenable, but we have many structurally enabled target systems in the world to deal with already. So that's not a huge concern. Now, yes, once we have determined by using this method what the hits are, it certainly helps to alleviate what is the next path forward in terms of a biochemical or a biophysical assay, because then you can just limit the number of compounds you need to try setting up biophysical assays on. The biophysical assay, setting up an SPR is not trivial either. You need to make sure the protein is properly immobilized on the chips. They have to not precipitate on the chips. They have to give you a good reading. And so on. And then if you're working with a target for which there are no hits ever before, it's a novel target, even standardizing the assay becomes a problem, right? So starting with the known hits that you know are directly binding helps you to solve all those problems and reduce labor, effort, and going down the wrong rabbit hole in these processes, for sure.
Okay. Thank you. I have a follow-up question just because you mentioned it's not possible to get crystals for every target. So how often would you say, in your experience, you have been unable to generate the crystals versus have been successful?
Yeah, so that's a very good question. So as I mentioned, we've been doing high throughput structural genomics for 15, 20 years. And we find, and during that time, we were working on all novel proteins. So these were proteins that had never been crystallized before. Their most similar structure in the protein data bank was probably at the most 20%. So we would go for really remote homologs. So we have done this characterization over 15 years, and we were solving at our peak. We were screening 800 crystals a week at the synchrotron, collecting and solving 200 novel structures deposited in the protein data bank every year. So we've done a huge amount of experimental work on this and determined that the success rate of a novel protein altogether, if it has never been done before, is still fairly low. It might be around 20%, 18, 20%. However, in standard drug discovery in industry, people are not usually working on proteins that have never been crystallized before. So when we look at our success rate in terms of working with targets for which structures have already been generated, or there's a structure in the protein data bank, or crystallization conditions have been looked at before, our success rate in trying to reproduce that protein from literature, from generating crystals, is about 95% or more. So we can really, generate those, and in many cases, we've actually optimized those crystals far more than they were reported in the protein data bank or published conditions. So, for example, in a recent SARS-CoV-2 target that we were doing, the published structure is at 2.9 angstrom resolution, and we got it on a 2 angstrom resolution, and we've done this for multiple targets. In one case, a structure has been published, many publications on it. We now have the highest resolution crystal structure, 1.5 angstroms, with very nice solvent structures that we can use for drug discovery to displace water molecules and build out the compound. So in most cases, we actually have a very high success rate for reproducing them, as well as further improving their diffraction qualities and their reproducibility to be able to run these screens, because you cannot run a screen with crystals that are not reproducible, right? So we have to spend quite a bit of effort in trying to make sure that if we set up 100 crystallization drops, we'll get close to 100 crystals or so. Or if the crystallization reproducibility is low, then we need to scale up crystallization to accommodate for that. So typically, it's pretty. As I said, in 11 targets that we have done so far, we were able to produce crystals for all of them, and that has not been such an issue.
Great.
Part of it comes because we do have a lot of background in trying to work with crystals and how to get them to be better, how to optimize our data collection strategies at the synchrotron using different kinds of synchrotron beam capabilities to be able to get good data. We collect data from bad crystals, poor crystals. So there's a lot of crystallography art that goes into good data collection as well. So I think that helps us a lot as well.
Great. Thank you very much.
Thank you again, Dr. Debanu, for your great presentation. And with that said, we'll be heading to the next presentation by Sadiq, Director of chemistry from Terramera, based in Canada, and he's going to be giving a talk on discovery of synergy.